Thank you very much for inviting us to present our work. It is a great pleasure to present our data, and we hope that it will help, that it will contribute towards standardization of metagenomic measurements in the human microbiome field. So as you mentioned, I am a member of the National Institute of Advanced Industrial Science and Technology in Tsukuba, Japan. So for today's agenda, I would like to briefly, after a short self-introduction, talk about microbiome community measurements by metagenomics, and especially stress the need for standardization. Then I will briefly introduce the Japan Microbiome Consortium, which has been the main driver of the work I am presenting today. Then I will present a short design and result of this study and summarize and look at the future perspectives. So as a short self-intro, so I'm a member of the Biomedical Research Institute in AIST, Japan. So our group is a microbiome center. So we are interested in developing state-of-the-art tools for multiplex biomeasurements of complex microbial communities. So basically, at the current stage, this is focused on next-generation sequencing and its application to human microbiome measurements. So specifically, we are interested in developing and applying experimental and computational tools for metagenome scale biomeasurements. And as Christine mentioned, we are mainly focused nowadays on development of materials and methods for quality control and standardization of the measurement methods. And we are also working with industrial partners and other institutes towards the development of techniques for microbiome control based on some of the research we are doing in our lab. So let me briefly start. I think most of you are familiar with how a metagenomics analysis pipeline looks like, but I would like to just briefly summarize it to get us on the same page. So it is a highly complex workflow to go from sample collection to final results. So it starts with sample collection and storage. Then the DNA or RNA or DNA is extracted from the samples. The sequencing libraries are prepared. This can be either amplicon sequencing or shotgun sequencing libraries. Then finally, the sequencing is performed. And after that, the data is processed, which include quality trimming, etc., or removal of poor sequencing reads, and finally, data analysis. So at each of these steps, there are lots of experimental choices that we can make. So, and this introduces a lot of variation in the measurement. So depending on each experimental variable that we choose or computational variable that we select, we will get a different outcome of the measurement. And there is, of course, no unbiased method. So each method has its own bias, and this is typically largely unknown. And as a result, there is generally very poor reproducibility and comparability of measurement results that are generated by different laboratories or in different studies. So, and this is shown in the PCA plot. This is data from the MBQC project, where they looked at a large set of variables showing that all of them, and mainly DNA extraction, are responsible for large variability in measurement results across methods and accordingly across laboratories and studies. So when we think about standardization, so this really stresses the need for standardization. At present, it is very difficult to compare data sets that are generated using different methods, each with their own largely unknown bias and errors. So this stresses the need for standardization. And here, when we think about standardization, we typically consider three main topics. This includes reference materials. So this can be either mock communities, which are mixtures of genomic DNA or cells of around 10 different microorganisms with a known abundance and known composition. Then more recently, fecal samples have also been more frequently used as reference materials because they are fully representative of the samples that we are interested in. Another component is validated methods. There are no standard methods at this time, so I refer to validated methods as methods that have been shown to have a good performance as tested using known samples, known reference materials. And also the last component is interlaboratory studies, where we can look at either what is the variation introduced due to different methodological variables, or what is the proficiency of different laboratories. So as Christine briefly mentioned, the work I will be presenting was started by the Japan Microbiome Consortium several years ago. So this is a consortium that was founded by major industrial companies in Japan. And their mission was to promote and accelerate industrialization of human microbiome research in Japan. So this is a list of the companies. This is not exhaustive at this time, but I just want to highlight there is a large diversity of companies that are involved in this consortium. Of course, we have the pharmaceutical companies and food companies, but we also have industry related to chemicals, biotechnology, etc., that are all now interested in leveraging the human microbiome to develop new products or services. So, and this is a slide showing the roadmap and main activities of the Japan Microbiome Consortium. So the shared pre-competitive goals of this consortium was to develop microbiome community measurement standards, which is the main topic of my talk today. And this will include developing of standard operating protocols and also providing more community reference materials to the industry. And using these standard operating protocols, we also have analyzed a cohort of about 1,000 healthy Japanese people. So this is to gather baseline data on the gut microbiome in Japanese individuals. And this can then be leveraged by different companies to compare to specific disease cohorts and even identify targets for developing microbiome products. So the activities are quite broad. So the main one is, of course, development of recommended protocols and related guidelines for microbiome analysis, and development of a microbiome database of healthy individuals. So a main aspect of is all the data that is generated is freely shared within the consortium, and the healthy cohort study will also be published in the future. So will this be available? And because many companies involved in the consortium are relatively new to the microbiome field, we focused a lot on exchanging information and activities between the different organizations. So broadly to improve human microbiome research and development in Japan. So most of the data that I will be presenting today has been published several months ago in Microbiome Journal, and this is the headshot of the manuscript. So please feel free to download this and have a look at it. It has some more detail than what I will present today, but the main message is the same. So the study design. So we were looking at mainly methodological variables at the stage of DNA extraction and shotgun library preparation. So at this step, we skipped sampling procedures and storage conditions, although we recognize this is important. So for the DNA extraction kit, we looked at 9 different kits and protocols. And for NGS library construction, we analyzed 12 kits. And this was done in phase 1 of this study. And this led to the development of standard operating procedures based on protocols that were highly performant, as evaluated using the mock communities. And based on these SOPs, we then analyzed the intra-laboratory precision of the SOPs and also the inter-laboratory variability of the SOPs. This was done by an inter-laboratory study involving 10 laboratories from the industry in Japan. So for phase 1, we mostly used mock communities. So these were newly developed DNA and cell mock communities. So by comparing the results to so we assigned the ground truth value to the mock communities and then assess the performance of the DNA extraction and library construction kits based on their agreement to the assigned ground truth. And those mock communities were also used for evaluating interlaboratory and intra-laboratory precision. And for that part, we also used fecal samples. So we had 5 fecal samples that were sent to different laboratories in addition to the mock communities. And so the mock communities were newly developed in this study, and this is a list of the bacteria that are present in the mock community. So most of them are from the human gut, but some of them are also from skin. We generated complete genome sequences for all of the strains, for at least for the strains for which no genome sequences had been available. And just as a technical detail, for the assignment of the abundances of the mock communities, we used total DNA concentration measurements. So for the DNA mock, we used fluorometric DNA quantification. And for the cell mock, we used a method that was recently developed that used acid lysis of the cells and then HPLC to determine the released adenine. And this allows us to quantify total DNA content in the cell cultures and allows us then to mix the mock communities in predefined proportions. So just briefly, the GC content varied from around 30% to 60%. So this will capture most of the variation in GC content in the human gut. And the cell mock community had a range of different gram-positives, which are known to be more challenging to extract DNA from. So those two mock communities should be appropriate to challenge the different methods. So let me now jump to evaluation of sequencing library construction protocols. So we evaluated this list of protocols. So they were all commercially available protocols. Several of them used enzymatic fragmentation and others used ultrasonication using the Cuvaris instrument for DNA fragmentation. All the kits were evaluated using PCR-dependent methods, starting from low input DNA or high input amounts of DNA. And a set of those kits was also evaluated in PCR-free format, starting from a higher amount of input DNA. And the one-letter abbreviations are the ones that are used in the manuscripts and throughout the rest of the presentation. So this is an overview of all the data for the library construction. So each facet is the result of a different strain, and it shows the relative abundance in the mock community measured using the different protocols. And generally, we see that the variation is relatively small. And if there is variation, it tends to be for strains with a high or low GC content. So this is apparent by realizing that the facets are sorted from low GC content to high GC content. So we see more variability at those for genomes with a high or low GC content. And this is shown here where I plotted the CV or the coefficient of variation of the measured abundances for each strain across all the different methods. So we see that the most methods yield consistent results for a GC content around 40 to 55, but beyond this range, several methods lead to more variable results. And this was impacted by the number of PCR cycles used for library amplification. So for the highest number of PCR cycles, starting from 1 nanogram of input DNA, we see larger variation across the different methods. And this is just showing what is the trend of the effect of genomic GC content. So we see for some methods, we see an overrepresentation of low GC content organisms, or for other methods, we see the opposite trend. And also we noted that for some of the kits that had a higher GC bias, and those used PCR amplification, we see, of course, a higher number of PCR duplicates. So the best practice here would be to reduce the number of PCR cycles, or if possible, omit PCR amplification of the libraries to to prevent any GC bias that may be introduced. So, and based on the known composition of the mock communities, we next sorted the different kits in terms of performance. So in the left plot, this is shown as the adjacent distance, which is a distance metric that is useful for compositional data. So, and they are sorted from top to bottom. So the higher adjacent distance is more deviation from the assigned ground truth. So, and on the right-hand plot, we have the absolute fold difference of each strain compared to the ground truth and also the geometric mean of this. So this allowed us to sort the best performing protocols, and this formed the basis for our SOPs. So we selected two different protocols. One is the QIAsec-FX DNA library kit from QIAGEN, and this uses enzymatic DNA fragmentation and can be performed with PCR or without PCR for library amplification. And then we also recommended Protocol K from Takara Bio, the smarter Truplex DNA sec kit, which uses physical DNA fragmentation So we of course looked at some other aspects of the protocol performance. For example, we can see that, as expected, the enzymatic methods have a slightly higher fragmentation bias, which can be seen as the uneven distribution of bases in the beginning of the reads. However, this fragmentation bias has minimal impact on the accuracy of quantification as evaluated using the mock communities. And also we noted that some libraries with longer fragment sizes tended to give more continuous assemblies. So this is one of the reasons that we selected the Protocol K or smarter Truplex as an additional protocol in addition to the QIAsec-FX, because we noted that the QIAsec-FX in some cases in our hands led to shorter fragment sizes, which led to more fragmented assemblies. So depending on the application, it's beneficial to choose a different protocol in some cases. So in terms of protocols for DNA extraction, so this is the list we used, or the set of protocols we evaluated. So the main point is that all of the protocols we used used bead beating for cell lysis. So we did not evaluate any enzymatic methods or methods that only use enzymatic means for cell lysis, because I think it has been well demonstrated in past literature that bead beating is necessary for recovery of gram-positives in fecal samples. So all of the kits use the FastPrep 24, except for Magatrack Power Microbiome kit, which performed bead beating in a 96 well plate format. And one of the kits we also evaluated was IHMS Protocol Q, which was developed by the International Human Microbiome Standards Consortium several years ago, and which has become one of the standard or one of the validated methods for DNA extraction from fecal samples. So this is a slide showing the performance of the different DNA extraction kits. So as shown in the gel, all of the methods yield high molecular weight DNA that is suitable for metagenomic analysis. And then looking at the abundance of the different species separated as gram-positives and gram-negatives, we can see that Protocol O, which used bead beating in 96 well plates, was slightly less effective in recovering gram-positives. So when in our hands, the bead beating in plates was less efficient, and so we are not proceeding with that method, although it's higher throughput. And also we see that by increasing bead beating time, we are able to recover more of the gram-positives. So this is shown in this slide. And also we see that our protocol, at least three 1-minute cycles of bead beating yields a total abundance of gram-positive that was comparable to that of Protocol Q, which is the IHMS protocol. And on the right-hand side, we see as an example, for protocol N, we see that bead beading increases the recovery of the gram positives specifically, while the abundance of the gram negatives is less affected, which is the expected trend. So as for this DNA mock communities, we next rank the protocols based on their agreement to the expected composition or ground truth. So as I already alluded to, protocol O, which uses bead beading in 96 well plates, was less efficient in recovering gram negatives, and less efficient in recovering gram positives, and thus had a poorer agreement with the ground truth. So, and the highest performing protocol is protocol N, which has the best agreement to the ground truth. And for protocol N, we recommend a bead beading time of at least three times 1 minute, or 3 rounds of 1 minute bead beading. And this is able to recover most of the gram positives that are known to be present in the mock community. So based on this, we had a set of standard operating protocols for library construction, so protocol K and protocol B. And for DNA extraction, we had protocol N. So we next evaluate the intermediate precision of those methods. So intermediate precision refers to the variation within a single laboratory due to varying operators, reagents, lots, etc. And also we set up an interlaboratory study involving 9 different laboratories in addition to our laboratory. And those were laboratories from the industry in Japan. And they had no specific experience with the SOPs and just followed the recommendations that or the protocols that we shared with them. So this is the results of the intermediate precision and interlaboratory reproducibility of library construction. So on the left-hand side, we see a plot of the pairwise Bray-Curtis dissimilarities of different pairs of measurements. So smaller Bray-Curtis dissimilarity means that the measurements are more similar to each other. So we see that for both within a single laboratory or across laboratories, we have very small differences in measurement results, almost comparable to that of technical replicates. So this suggests that library construction using both protocol K and protocol B is very reproducible across laboratories. And in terms of CV, so the coefficient of variation of the abundance of different strains is generally less than 5% across laboratories. So this means very high reproducibility. And this is the result of intermediate precision and interlaboratory reproducibility of DNA extraction as evaluated using the cell mock. Here evaluated using the cell mock community, yes. So we see that again, across different laboratories and within a single laboratory, variation is very small and much smaller than what we observe across different methods. And also in the bottom hand of the left-hand plot, we can see that the custom methods evaluated by some of the laboratories yielded more different results, so which is expected. And in terms of the CV, again, it's within a laboratory, the CV of the abundance of different strains will be less than 5%. And across different laboratories, it should be around 10% or less. So again, the very high reproducibility of DNA extraction following a single protocol across 10 different laboratories in this case. So, and this is evaluated using the fecal samples. So we evaluated 5 additional fecal samples. So we can see again that within a single laboratory, measurements of the fecal sample are very reproducible. Across laboratories, there is some increased variability with 2 outliers in this case, but still the variation is very small and much smaller than the differences between different methods or with different custom methods used by some of the laboratories, and also much smaller than the differences between fecal samples from different individuals. So, and in terms of CV, the CV of the species abundances is around 10% for within a single laboratory and around 20% across laboratories. So I think this is quite acceptable at this stage of standardization efforts. So next, we also evaluated our methods in the context of the Mosaic Standards Challenge. So this is an international interlaboratory study that aims to look at the main sources of variability in the metagenomics workflows. So the set of samples that is included in this challenge kit are 2 DNA samples and 5 fecal samples. So we evaluated 3 different protocols. One is protocol P, which uses phenol-based DNA extraction methods, which also performed well in phase 1. Then protocol Q, which IHMS protocol, and protocol N, our SOP. And for library construction, we used the QIAseq-FX protocol, and our sequencing was performed on an NextSeq 500 instrument. And this is the results for the 2 DNA samples that are included in this challenge kit. So we can see that there is a relatively small variation due to differences in library construction, although we do see some laboratories that yield different results. But our protocol BL or the QIAseq-FX yields results that are highly consistent with that of other methods. That this was true for both of the DNA mixtures that were provided. And this is the result of the fecal samples. So our protocols tended to give a slightly higher amount of gram-positives than the other protocols that were evaluated in the challenge. So this seems to suggest that our protocol is very effective in recovering gram-positives, which is consistent with what we observed for the CELMoC community evaluation. So, and this is also shown here, our kit. So these box plots are the results of that are publicly available on the Mosaic Standards Challenge website, and the symbols are our data. So we are able to recover a higher diversity of species in terms of Shannon diversity. And also our method is most comparable to protocol Q compared to the other methods that were evaluated by other participants. So, and based on these SOPs, we also try to set some target values for achievable performance. So it is always challenging when you analyze, for example, a mock community. What is the acceptable level of error that would be permitted for a method to be deemed acceptable? So this slide is just showing, we set some implementation guidelines or some guidelines on how to implement the SOPs and which acceptance criteria to implement. For example, when you evaluate a new method using the DNA mock community, we suggest that the differences to the expected value should be less than 1.15-fold on average across the different strains. And for the CELMoC community, we are allowing a difference of 1.55-fold compared to the assigned ground truth. But I should note those values are specific for this specific mock community, so they may have to be re-evaluated when using a different mock community. So based on this, I would like to summarize our study. So this was started by JMBC, which is an industry-led consortium with shared pre-competitive goals. So what we achieve is development of DNA and cell-based mock communities that will be available to the microbiome community through the Knight Biological Research Center in Japan. Let me mention here that the mock community we used in the study that I presented was version 2. So we now developed a version 3. So 2 different mock communities, and this validation data of this mock community will be hopefully forthcoming in a journal publication in the next months. So using these mock communities, we performed a comprehensive assessment of the impact of experimental variables at all steps of the measurement workflow. And this formed the basis for the development of the SOPs. And as I mentioned in the beginning, we adopted the SOP for the analysis of a healthy cohort of about 1,200 healthy Japanese individuals. So, and with that, I would like to acknowledge everybody who was involved in this study, from JMBC, from RIKEN, from NITE and AIST, and also the funding agency NEDO. So I still have a few minutes left, so I think I would like to take the opportunity to also present some other work we have been doing related to standardization, specifically development of synthetic DNA standards for microbiome community measurements. So this is a study we published several years ago where we developed a synthetic variants of 16S rRNA genes. So those consist of conserved regions or portions of the 16S rRNA gene that are common, that are from a natural sequence, for example, from E. coli. And then we replace the variable regions of the gene with random sequences. And those random sequences are uniquely identifiable because they don't share any similarity to sequences in public databases. So these standards can be amplified using any PCR primer that will bind to the conserved regions, and then can be detected in any sample based on their unique random variable regions. So, and just briefly, we developed a mixture of 12 different types of synthetic 16S rRNA genes. So this can then be added to the DNA, to the sample, either at the point of DNA extraction or at the point of library amplification or library construction. And then this is a typical quality control plot where we plot the number of sequencing reads versus the spike in amount that has been added. So in this case, we see strong linearity between both. So this suggests that this measurement was performed well. And with that, I would like to thank you for your attention. And again, I would like to thank QIAGEN for giving me the opportunity to present our work. And I would be happy to answer any questions that you may have.
QIAGEN.
Sample to insight.